Okay, so let's pick up where we left off on the last lecture. We were talking about chemical building blocks of life and different groups of macromolecules that are out there. And we were talking about carbohydrates. Key goal or the key function of carbohydrates is to provide us with energy. We talked about monosaccharides, polysaccharides, disaccharides, and we're leaving off with the polysaccharides. We left off there. Again, keep in mind, starch is a plant-based polysaccharide, same as cellulose. Both of those are complex carbs. The human body cannot digest cellulose, so we use it as a form of roughage. So you do want cellulose in your diet. This is your bran, cereal, bran, things of fiber base. Glycogen is an animal-based polysaccharide that we can digest. We store this in our liver and in our muscle tissues. So when your body needs energy, it goes to your stomach first, your bloodstream. If it still needs energy, it's going to go for the carbs that are stored in your liver in the form of glycogen. Pull that out and start breaking it down and digesting it down to glucose. Glucose can then enter into the cell and get converted into ATP. Chitin, on the other hand, found in the exoskeletons of things like grasshoppers and crickets and shrimp and lobsters, those types of animals, is also a component of the cell wall of fungi. It's one of their defining features. We can't digest it. So if you eat a grasshopper or you were to eat a crab leg and get a little piece of the shell, it's not going to digest. It's going to pass through your system. A basic tenet about food: if our body cannot use it and it cannot store it, it gets rid of it. It excretes it out. And with that idea in mind, this moves us into the artificial sugars. So all sorts of products are out there that are diet Pepsi, Diet Coke. These are things that contain artificial sugars. And the reason why they work is that your body can't recognize it. So natural sugar, something like sucrose, it's a disaccharide. 2 glucose molecules bonded together. Your body will recognize it, it will chop it in half, take each glucose molecule, move it into the bloodstream, move it into the cell, and use it to make ATP. Now, on the right-hand side here, we have sucralose. This is commonly marketed as Splenda. And there's a bunch of them out there, and they're all very similar. If you notice, the difference between Splenda and natural sugar is the chlorine. So chemists have removed the OHs or hydroxides from natural sugar and replaced it with a chlorine atom. This makes the molecule stable, but it's something the body doesn't recognize. So it takes very little of this sucralose or Splenda for our body to actually detect it. It's about 600 times sweeter than natural sugar. This is why if you cook anything with Splenda, you use hardly any, teaspoon, where if you were to cook a similar food, maybe you're baking cookies or making a cake, if you were to use natural sugar, you might use half a cup or a full cup. So the ratio is significantly different with artificial sugars because how much sweeter they tend to appear or be, how our body reacts to it. But the thing is, when you eat the artificial sugar, about 80% of that is excreted out of the body because your body doesn't know what to do with it. The 20% that's absorbed, that actually doesn't even get metabolized. It kind of rolls around in your system for a while, and your system doesn't know what to do with it. This is why you can drink Diet Pepsi, and it has 0 calories. So when you have Diet Pepsi, you're getting caffeine, artificial flavors, you're getting the carbonation, the water, but you're not getting calories because it's sweetened with these artificial sugars. So just keep that in mind. When you put something into your body that it does not recognize and it can't digest it, your body wants to get rid of it. And it tends to come out through urination, defecation, or if it's really bad, your body may reject it quickly and now you're vomiting. So, all right, again, keep in mind, carbs are the primary source of energy that we want to be using when we eat. Okay, now we're going to take a look at a second group called the nucleic acids. Nucleic acids are made up of a subunit called the nucleotide. Now the nucleotide is a 5-carbon sugar, which we can see down here. The 5, 4, 3, 2, 1 represent carbons. This is a sugar. It's known as a pentose, or 5-carbon sugar. It has a phosphate group attached to it, and then it has a nitrogenous base. So we'll revisit nucleic acids when we get into genetics and DNA. But the easy way to remember this, you got a sugar shack, a phosphate chimney, and a porch that's made up of nitrogen. Now the nitrogen base porch, there's only 4 different options when we talk about DNA. You have an A, a T, a C, or a G. Adenine, thymine, cytosine, guanine for your nucleic acid. When we look at DNA, those are the nitrogenous bases. When we look at RNA, we have an option called uracil. RNA doesn't like thymine. Instead of using T porches, it uses uracil for porches. So we'll come back to nucleic acids and talk a lot more about those as we get into genetics and molecular biology. Nucleic acids are the fundamental piece of information that carries a code for all life. DNA is a double helix, which you can see over here on the right-hand side. RNA is a single strand of nucleotides, and then we have ATP, which is also fits under the category of nucleic acids. Now ATP, sugar shack, nitrogenous base, adenine, it's got the A porch, but ATP has 3 phosphates, 3 chimneys. That's what makes it such a high energy molecule. That's the one we want to remember when we talk about energy. ATP. Incredibly important because that is what all life depends upon when we talk about sources of energy. Okay, again, we'll revisit nucleic acids a lot more when we get into genetics and DNA. So our third group of macromolecules are the proteins. Now what I want you guys to do is fill in the blanks here. What are the roles of proteins? Proteins are actually the most diverse group of macromolecules. They serve more roles than any other group. More roles in DNA, the nucleic acids, more roles than the sugars and the carbohydrates, and more roles and functions than the lipids. Proteins are absolutely essential for life, and we have to make sure we get the right ingredients to build proteins in our bodies. So you look at the images here. Proteins make up muscle tissue. So movement, that's based on proteins. Proteins make up our hair. Structural support, made up of protein. Proteins help with enzymes to allow reactions to proceed at a faster rate. Your immune system is made up of proteins. They transport things. So over here we're looking at hemoglobin. This is a protein-based structure that carries oxygen. Proteins also help in regulation. Hormones that determine the growth, the development, how our body functions are all protein-based structures. And then storage. Proteins are often used to store things. The albumin in eggs, that kind of white slimy stuff, that's all protein. Eggs are a great source of protein because they have a lot of protein within it. Now, protein is built, all proteins, no matter what organism we're talking about, plant or animal or anything, proteins are built from these 20 different amino acids out there. So there's a standard 20 amino acids that the vast majority of life use. For humans, 20 amino acids. So focus on numbers 1 through 20. Proteins are then built out of different combinations of those amino acids. What makes each of us different is simply how we arrange those amino acids to form a protein. And a very important rule to remember about proteins, shape equals function. It's one of the themes we talked about. Shape of the protein determines the function of the protein. If you change the shape, you change the function. And if we're talking about something where we should have a certain shape, maybe a primary structure or a secondary structure, and that shape changes, that protein cannot do its job. And that can lead to significant problems. Imagine it's a pathway that requires a protein to function. You change the shape, protein doesn't work, pathway shuts down. So the levels of complexity of protein: primary, secondary, tertiary, quaternary. First, second, third, fourth. Each level, as you go up in levels, it increases in complexity, and is typically built from the previous level. So the first level, primary structure, is just a string of amino acids. Secondary structures, those amino acids strung together and then folded or twisted into this helix shape here. The third level, basically you take several of the seconds, combine them together to make the third. To get to the fourth level, you take a bunch of these tertiary structures and push them together to make the fourth level structure. So the complexity increases as you go up. Now the job of our digestive system is to be able to digest this. Eat a piece of meat, might be eating a third level protein. The digestive system has to chop it up, bust it apart to the second level, bust it down to the first level, and then eventually bust it down to individual amino acids. So proteins are incredibly important and built in 4 levels of complexity. Each level dictates what it can do. Now, when we look at proteins, sometimes they come in what we call motifs. These are 3-dimensional structures of proteins. They often form tubes or certain shapes. So some parts of the cell might have motif protein structures that allow things to move in or out of the cell. Proteins can also be in a shape known as a domain. This is a substructure within the tertiary structure. So here's our tertiary protein, third level. The domain is an individual shaped protein within the larger structure. Now if you change this domain, it will influence domain 1 and domain 2. So again, the idea here is looking at the complexity of these structures, and that they all have to be shaped specifically to work in a certain fashion. And if that shape changes, we have significant problems. Now there are proteins out there known as chaperones. Chaperone proteins actually help other proteins form the correct shape. So a chaperone up here will take a protein that's misfolded, bring it into the chaperone, use some energy, put the little cap on, rearrange it, kind of twist it around, reform it, shift it around, and then pop it out, and hopefully it's refolded the protein into the correct shape. Otherwise, this misfolded protein up here will never function, will never be able to do its job correctly. So chaperones are an important protein to help fix bad proteins. So hopefully proteins are all working correctly. If they misfold, they're what we call a prion. There's different diseases out there. Chronic wasting disease, which is a huge issue with the deer herd in Illinois. Mad cow disease, Kuru, things like that are all due to proteins that misfold. But sometimes proteins get denatured. This is when their shape changes. Denaturation typically occurs because of a change in temperature or a change in pH. Those are the primary factors that shift the protein's shape. So when you denature a protein, you change the shape, you often shut down the protein's ability to function. Now sometimes the shape can be renatured, it can go back to the original. Other times it's just done and the protein will never work. So if anybody's ever gotten a perm, consider perms. You take hair, which is protein-based, you break the shape of the hair, so you're denaturing it, and then you're folding it back into a new shape and trying to reset it in that new shape. You're renaturing it in a different shape. So perming your hair is working with protein structures.